Swayam Prabha. Digital India. Educated India.
Introduce Dr. Rashmi P. Mohanty, Assistant Professor in Department of Chemical Engineering and at the Indian Institute of Technology, Madras. Her research lies at the intersection of chemical engineering, synthetic biology, biological engineering, and pharmaceutical sciences with focus on developing advanced disease models and drug delivering systems. Before joining IIT Madras, she worked as postdoctoral associate at the MIT, contributing to research in stem cell engineering and vascular organoid formation. Through her work in synthetic biology, disease modeling, and biomedical engineering, she continues to contribute significantly to healthcare and scientific innovation. We are honored to have you here with us today. Please join me in warmly welcoming Dr. Rashmi.
Yeah, thank you so much. So you already found something about me. Okay, thank you. So now probably we can start. So yes, so my background is mostly I finished my chemical engineering, my bachelor's and master's in India, and then I went to the US for my PhD, and my PhD was in pharmaceutical sciences. And after that, I did my postdoc at MIT, which was in biological engineering. And then right now I am back here in chemical engineering department again. So I will be giving one more talk. So today we are talking more about the drug delivery. So I will be giving one more talk which will talk about, which is going to be more about synthetic biology, what that field is. So today we'll have like nothing about the synthetic biology aspect. But basically, when I told you about my journey that I started as a chemical engineer, now also I am in chemical engineering, but in the middle I spanned in, between multiple different departments, right? So that's something that I found while I went for the interview with some of you. Like we asked this question that which is your favorite subject? Mathematics, physics, chemistry, biology, and then we made sure to ask you why you like that subject. And then some of you had the logic, and some of you had this question about I have multiple interests, and I feel bad that maybe we need to be more directed towards one, so that we are very targeted and we are learning something in much deep in one concept. So that's something that we will talk about through this one one field, that how interdisciplinary approach is necessary. So having multiple interest is absolutely necessary to come up with better innovations. So what we'll do is, like when I am saying drug delivery, it seems something more about like what is coming in your brain, in your mind in terms of just subjects. Mhm. Exactly, biology and anything else? Why chemistry? Okay. So, uh-huh. Okay, so the drug itself can be a chemistry molecule. Oh, that also you know. And what else is coming in your mind? Okay, so as rightly pointed out, so you are looking at drug and you are thinking about medicine, right? So that's why you are thinking about somehow there is some medicine which needs to go into your body, and that's why it has to do with, and as rightly pointed out, that you will have these molecules which are the ingredients that is going to treat you. They have chemical structure, so they are chemical compounds. But not only chemical compounds, there can be drug molecules of different, like not just chemistry, but then there can be some other type of drug compounds which we are not going very deep into it. But the point is, you brought about chemistry and biology, but I am writing that engineering principles, right? That how you need engineering, because we are not thinking about the last part, which is delivery. So that's what we are going to focus more today, not the drug part, but how to deliver the drug inside our body. Okay. So these are the learning outcomes. So I hope that by the end of the lecture, that you will be able to do this. So the first point is you will list some of these predominant routes of drug administration, because as I mentioned, we are not talking about drug in itself, but how we are going to. So when we take the drug molecule, then what are the different ways in which you can take it, and then how you are going to deliver the drug molecule at a certain location. So wherever your disease is, you want that active, the chemical compound, you want it to go and then treat wherever you have the disease, right? So, and then the second one is we will talk about something. So I wrote this very heavy term called pharmacokinetics properties. So that is something that you will come across. So which means majorly that once you take out, like, that chemical compound, then your it is going to interact with your biology. You brought about the biology. Now what that biology is going to do that drug molecule, right? So when you have a chemical compound, how your biology is going to interact and how that is changing, how that chemical compound is going to be changed or treat your body. So that is something comes under pharmacokinetics. So that term is coming from pharmaceutics. So that's why when you said already, like chemistry, biology, another field that is heavily involved is pharmacy. Okay. So now next what we will talk about is we will talk about some of these design principles, because we already talked about that how your biology is going to impact your chemistry molecule. But after that we will talk about, once you understand that how my biology is going to impact my drug, can we design drugs so that we will take advantage of the biological system, and then we will come up with more efficient systems so that you are getting faster treatment responses. Okay. So what are some of those design principles that are involved? So that is, you will see that many different type of engineering and also other type of like disciplines, fields are required to answer that question. And then finally, of course, we will kind of evaluate what are all the interdisciplinary, like how this field is interdisciplinary, and what are all the fields that are involved in this. Okay. So now coming to what are all the ways in which we administer therapeutics in our body. So now just think about any time you have taken a drug molecule, like a medicine, and what are all the ways in which you took it. What? Injection, capsules, tablets, oral, syrups. What is? Oh, topical, IV, yeah. So broadly, these are the ones, right? So I mean, I will also summarize it in that way. So we'll talk about oral, but then when you told capsules, tablets, or even syrup, all of that we want to take in orally, and that is it's called oral route of administration. And then you can take you can take it in multiple ways. Your tablet can contain some of these tiny particles, it can contain powders, and also you can take it as a solvent, right, as syrup. And then you also talked about intravenous, so IV someone already told. And then the injection, like you are taking it, you are injecting it directly into your vein, so that's why it's called intravenous, into the vein. The next one is intramuscular. So you told about injection, and then this is the most common one that we know, that we are taking injection into our muscle. So and then the transdermal. So someone already said about topical. So that means we are thinking about, you have a patch, a skin patch, or you are taking ointments and applying on top of your skin, all this pain relief medicine and all. But also still, if I don't know if you all are aware of it, so nowadays there are these diabetic needles. So you can even take it to the skin. It doesn't have to go to your muscle level, but you can inject it to your skin. And of course, and also there are some of these, like when we are saying the patch, it can also be in terms of injections, but then micro injections, multiple needles which are forming that patch through which you are taking your medicine. And then this one, I think no one mentioned. So like the pulmonary, so if people have asthma, then they just take this inhaler. So that means you have a powder, but then you are you have to swallow it very rapidly. So what happens, like how these are different. So this is not actually oral route of administration, even if you are taking it like that. So what usually happens is you really want the impact to happen to your lung very quickly. So instead of going to your digestive system, that is going to your respiratory system. Okay. Also, you all know about all these eye ointments, right, or eye drops. or even nasally, sometimes through the nose you take some of this. So if you are taking it through the nose, it can also go to your brain. So that can be one of the place where you want to reach. So basically, again, we'll come back that you have a tablet, simplified form, and then if you will open that tablet, then you have that chemistry, that molecule. So those molecules are called active ingredients. So when you are actually taking tablets, then you have, so these active ingredients, they are hydrophobic in nature, they are really insoluble in water, and they are not tasty at all. So very bitter because you are taking it from the plant sources. So what happens is when you are taking it, then you want it to be dissolved and then distribute through to your body. But the idea is you also want to like it. So that's why, but when you take most of the tablets and syrups, it tastes sugary because they give that sugar molecule so that you will be able to, you will like it to at least take it. But so that tablet, it contains multiple different things, and then it is packaged into that capsule. But there is this one, that chemical compound that we are thinking that it's chemical compound, but also you can have biological like molecules, which are called active pharmaceutical ingredients, which are only the ones which are responsible to treat us. Okay. So now we will understand, kind of that why you need all of these extra things and all. But basically what we want is that you are taking that tablet or taking any medicine, but it contains that active pharmaceutical ingredients and multiple other things, and you are putting it through different routes of administration to your body, and you want that to reach a particular location, right? So we have these different organs, and you want that where exactly you want it to be delivered. So that is what is called drug delivery, that you are taking it, so you are you are injecting it, but you want it to be delivered at a particular location. So that journey from site of injection to site of delivery, that is what is called drug delivery. So as I kind of pointed out, so with pulmonary, so why you are doing that, the compaction, like the asthma, the inhaler, is because you will reach to your lung very quickly compared to if you are injecting it even into your bloodstream, right? Because in your bloodstream it is going everywhere, but then you have the problem in lung. So it's much easier, convenient if you are putting something directly to your lung. Similarly to the brain, if you want to put something, then there is one line which goes from your nasal to your brain, or even eye to the brain. So can you take those advantages? So those are called routes of administration, and the aspect of drug delivery. Now I don't know if this is going to work. So this is a YouTube video short. So most probably no, because we are using this other system. So what happens is, so once you are taking the tablet, again coming back to the tablet, I told you that it contains multiple things, and then you have that active ingredient. So and also we talked about the digestive system and also the respiratory system. So it will go through to the digestive system, and then once it reaches the stomach, then it will be solubilized, and then once it's solubilized, then you have all this small intestine, right? So it will go to the small intestine afterwards, and small intestine is very, it has a lot of surface area. So it's a smaller tube, but it has a lot of surface area through which these active ingredients, they get absorbed through that small molecule, small intestine. And once that happens, then after that small molecule, small intestine, you will have your, it will get access to the vascular system, that means to the bloodstream. So that's why it needs to be solubilized first, then it needs to be absorbed by the small intestine. After that it will distribute, it will go to your bloodstream, and since you have blood everywhere, it will be distributed throughout the system. But what happens is, during that time, during that journey, so if you will see, we have some of these organs which are highly vascularized. So our liver has a lot of blood vessel, our heart has a lot of blood vessel, lung has a lot of blood vessel. So what ends up happening is, more of these ingredients, they will go to liver very quickly because they are near the small intestine, and then your liver is very close to that, right? And then your liver has a lot of enzymes, and its job is to do metabolism, right? So that's how we digest all our food, all the metabolic activities happening. that means your liver is going to create these metabolic products out of your ingredient. So then your entire chemical compound, if it's turning it into smaller parts, then that is not active anymore to treat you. So that's why, so after this metabolism, if it's converting it into tiny parts, then what can happen is your drug molecule can get eliminated either through the liver, or it will go to the kidney and then through the kidney. Okay. So you will see that that starts with ADME. So this ADME is treated as that pharmacokinetics parameters. So that means every time you are putting anything in your body, it needs to go through those different process. But again, we will come back to different routes of administration and all. But after this elimination, whatever is left around, that is going to circulate through the blood. This is happening simultaneously though. And then while circulating it, they need to find the exact target location, go there and then treat you. Okay. Any questions? Yeah, that is insoluble. But then you are putting, not the ingredient doesn't become soluble. So you are already giving, so what will happen, if you are just taking that ingredient, then we have a lot of immune system which is going to just reject them. So they won't even reach our stomach. So that's why you are putting this extra covering as the tablet. So you have a lot of other material which are called excipients, so that they are safely carrying your tablet at least until the stomach. Then that will get open and the tablet is getting solubilized, not your active ingredient. So at least your active ingredient reaches your stomach or small intestine, and after that they will get absorbed through the small intestinal wall. Yeah. Exactly. So that we will come back. So, right. So, okay. Now you tell me, why don't we just inject it into vein? Which one do you like better? When you have to take a tablet, or when you have to go to clinic and then inject it? So that is the point, that you always have to think about the patient compliance, if they are going to like it or not. Okay. And also when I told you about the diabetes example, I think that is the only majorly only situation where people are taking it by themselves, right? The injections. Most of the times either you are going, and for IV you are never doing it at home, you still are going to hospital, right? So now we'll just talk about that oral first, and then we will come to the other routes of administration. So why the tablet is going to be even dissolved? So the question is, again the tablet dissolving, not the active ingredients. So what happens is, if you will look at our entire digestive system, and you probably already know it, that there is different pH at different location, right? So that's why we know that our stomach has acidic pH, and then our small intestine has some other pH. So basically, so this is one of the very glimpse of, this is a design rule. What people do is, somehow they put these ingredients which is going to until the time they reach the stomach, they are not getting solubilized even if there is water, but only when they will see that particular pH, at that pH only they are going to dissolve and then open up. So this is a very small diagram, but then we will go through some other of these very complex design systems. Yeah. So, but you don't want that. But what happens is, so the tablet doesn't contain just that active ingredient. It contains multiple other parts, that as I told you. So they still can get solubilized. When your tablet is going slowly, then what can happen is, it's still in contact. Even when you swallow it, it's still in contact with water. You want that to be remain intact by the time it's reaching the stomach, reaching the near the small intestine. Then only, because until that entire time you want it to be protected, then only you want the solubilization to happen and then the ingredient to come out. So I gave you that as one of the example, but then as you told, so even when you are taking IV, okay, you talked about IV, but still you are not just taking the active ingredient. in IV also, so you take it with different type of formulation, even if it's a liquid system. Similarly with syrup also, so if you will think about how, like, when you are taking syrup, it's a viscous substance. It still has multiple different type of, like, components. So basically, like, it's not that you are not going to have enough immunity. You are right that in a stomach, sorry, in this system you are not going to have enough immunity, but that part think more about, like, you don't want early release of your active ingredient. But even when you are, so the immunity I gave you overall, any type of like, administration. So, no, I am saying that in stomach. So stomach has a way less acidic pH. Yeah, so basically, pH I am just giving you as one of the example and very relevant for the oral system, that how you want it to be. So it's not that like, biology is never 0 and 100%. So there can be still some early release, but then majority of it, because you are trying to design it in a way that you are targeting it, that at this pH level only I want most of my release to happen. Okay. Okay, so basically here is a very, like, very broad schematic, where what we are saying is you have a system where you have your active ingredients very compact, but then when you are changing the pH, then there is some swelling that is happening, and because of that you have release of your like, ingredients out of that system. So so what are the parameters that are determining this drug absorption? Because we talked about that you will have the absorption first. So one is, as we talked about properly, that you want the solubility, that it needs to solubilize properly. If you are getting better solubilization, then you will have more ingredients that is available. And then the other part is the permeability. That means you have your, like, you want in your intestinal layer, you have these cells, cell layers through which you want your active ingredient to pass through, right? So that is a term which is called permeability. What is the permeation rate? So these two are going to majorly decide that what is the absorption rate that is going to happen. Then once your drug absorption happens, then what will, okay, so I have all of them simultaneously. So basically your like, absorption again through the small intestine, it's going to your bloodstream, and then as I told you, it's going to circulate, like it will have access to the blood, and then all of these can happen. So you want blood to transport your active ingredient to the disease site, right? So that, so transport is a like, a very generic term, that you are just transporting something. But then since you already have blood flow through your heart is a pump which is pumping it, and because there is already some like, transport which is already happening because of the pumping action. So because of that, your whatever is your solute, it is going through that pumping action. So that type of transport is called as convective transport. So the transport has two, has anyone knew about this? So I am just giving you more, like, engineering terms. So transport has two ways. One is diffusion, one is convection. Convection is a term which you are going to use if you are putting some pressure to a fluid, and along with that pressure, when the fluid movement is happening, whatever your desired solute is, that is moving through that. Diffusion is a process where you don't have any like, any gradients, any pressure gradient, but just because you have higher amount of your concentration and lower amount of your concentration, so the movement is happening because of the change in the concentration level. So here, so because our heart is already pumping, our blood is already anyways moving, that's why our drug molecule is going to go along with it. And as I already, as we mentioned, a very significant thing is going to happen simultaneously, which is elimination. So to go little deeper in it, so our kidney, what it does, it can like filter anything that is beyond 5 nanometer. So let's say your liver is already metabolizing something. So After that, it is anyways going to, like, the bile duct and all. But also, if your active ingredient, if your drug system that you have, that is less than 5 nanometer, then very clearly you will see that it will pass through your kidney and then come out. So which is something that you don't want. So what is happening is you want higher absorption, but you don't want it to be eliminated. You want it to be circulated in your bloodstream for longer time, right? So that's why, that's what exactly, like, how does the drug, what is the available drug concentration in your blood? Because if it is circulating for longer time, then hopefully at one point it will reach your disease location. So what is happening is you have a rate of absorption, and then some of them it's like, you have absorption and you have elimination. So now we will plot a curve of concentration of your active ingredient in the blood versus time. Okay, so what is going to happen is, as we talked about, that if your absorption, when your absorption is happening, then you are going to have higher concentration with time, but with elimination it will go down. So now we will come to the point that when you are taking oral tablet, right? So until now we are talking about oral tablet. Then what happens is, after some time it's completely going down. So then what you will do, because you want the therapeutic effect, right? So if it is happening very suddenly, so you all do that, like, I mean, we all do that, not you all. So how exactly? Because if it is going down, that means you are not going to get the therapeutic effect. So can you think about how we will increase the concentration again? Exactly. So you will take another dose, right? So that is what is called the dosing. So you will take another dose, but now we will understand a little bit about that. So that is what. So basically, so there are these two layers of concentration. I am sorry that I have not written it properly. So the bottom layer that you see, this is called the therapeutic concentration. So basically, you want your active ingredient concentration to be higher than that, so that you are getting any effect. But then the top one that I put, if you have more than that, then you can have toxic effects, right? Because your blood is not going to directly take it to your site of wherever you have disease, or like, but anyways, in the second part you will learn about, we always try to do that. But the point is, if you will have very high amount of these active ingredients, they are reaching your safe location, healthy places, then they can damage your body at your healthy location, right? So that is something called toxicity. Are you getting my point? Okay. So this lower one is called efficacy concentration. Then the top one is called toxic concentration. So to get a therapeutic response, you always need to be in between that two range. So you always want to maintain your concentration in between that. So that's why when you will see that the concentration is going down, then you want to take another medicine, another dose of your tablet. And what that will do is again increase your concentration. So that's why when they ask you, the doctors are asking that you have to take medicine after each 24 hours, you have to obey that, because that's how they already designed it. So based on your age, based on your, I don't know what are the other physical parameters that they check, but then the dosing is already set in a way that if you are taking after every 8 hours or after every 12 hours, then only you are going to remain in that therapeutic window. Otherwise, either you are very low, like, you are not getting the efficacy, or you are reaching your toxicity level. Okay. So now, any question?
What is the term that is called when the concentration is below the therapeutic range?
Below the therapeutic range, I don't. I mean, I know that this one we call.
The above is
Oh,
toxic.
is toxic. That is just therapeutic. So therapeutic concentration, and then.
The lower limit of therapeutic.
Yeah, lower limit is therapeutic concentration. That one is toxic concentration, and then this one I don't even know if there is a term for that. Okay. So now we'll come back to the intravenous point, right? Because here we exactly the same point that someone already raised, that you are swallowing the tablet and then you are waiting because you want it to eventually go to your bloodstream, because that's where you want all the distribution and circulation to happen. Then why don't you just give it to your IV, the vein directly? So that exactly what happens, that you are just giving it directly to your vein. That means all your active ingredient is completely present in your blood. So that's why you are not worried about that absorption to happen. So so with our oral case, we saw that how it will go up and then it will go down, right? Because there is absorption and then elimination. But when you have your intravenous administration, then you are not wasting any time in reaching the higher concentration level, because you are anyways giving it directly to your blood. So that's why what you are going to get is, like, you already have your highest concentration here and then it is going down because of the elimination, but there still can be elimination because of multiple things. Okay, so that is the, like, most efficient way, but the problem is it's not convenient. Okay, so that's why whenever you need, like, a medical emergency situation, then only they will give you IV. Similarly, intramuscular, so what happens is you are taking it, so there are multiple layers. So we are also covering the skin kind of here. So you have multiple layers of the skin, and then you kind of have your blood, most of the blood vessels that is below your skin layer, and then you have your, muscle layer. So muscle also has a lot of blood vessels, and also it's much more receptive. If you are giving some of these ingredients, then muscle has, you can give little higher amount and all, but then there also there, there is access of blood vessel, but still it needs to penetrate some of these cell layers, tissue layers, and then reach your target site, which is the blood vessel. And then similarly the same is with the skin. So either you are giving, like, you can give multiple in multiple ways, but still it needs to penetrate through multiple barriers of skin and then reach your blood vessel. So you have something called epidermis, dermis, and then here are your blood vessels. So basically with that route also you have multiple, tissue layers through which your drug molecule need to penetrate and then reach your blood vessels. So with pulmonary, what happens is mostly you have it in powder form, so you are not injecting. So the pulmonary, has anyone ever used it, the inhaler? Okay. So it contains like very tiny powders. So when you are taking it, then it needs to, you want to avoid your digestive system, it needs to go to this. So your digestive system is at the front, your respiratory system is little at the back. So then the particle size should be such that, so these powders or these particles are directly coming and then settling down and then going to your entire lung. Okay. So the purpose of explaining all of this is that you can see that depending on where you are administering, you could see that there are multiple different biological environment your drug is going to see, right? So either tissue, either blood, either your digestive system. So then like just after this, let's discuss a few points and then we will come to the second part of the talk. So which route of administration that will go to bloodstream rapidly, I think we have, yeah. And the second one is, how would you rank different routes of administration based on patient compliance? Oral pulmonary, okay, scheme. Yeah, but also you need to remember that the pulmonary is very specific, right? You are not taking it to so that your your drug is going everywhere. But with intramuscular also, so when you take vaccines and all, so for vaccines you really don't need to reach a proper target. So there also you want your immune system to be developed. When you are taking something like an antigen, you want your immune system to be trained upon that. So that's why you can your muscle is more convenient, because it is going to be less painful compared to the intravenous route. Okay, now, so we talked about all of this. So this is what you already knew. That's how you take it. Now, as a patient, now, go ahead.
Let's say a person has an illness regarding their lungs. Let's say that they need to take a medicine, but it's very toxic to other parts other than the respiratory system. So how will you ensure that 100% or nearly 100% of the medicine will go to the coronary system instead of the digestive system?
Okay, now you tell me. But you want to take that only, the inhaler only? Yeah. So, what are some of the answers that is coming to your mind? Yeah, so that's why. So the design. So those for we'll cover pulmonary. But is anything coming to your or anything else coming to you? Hmm? Nebulizers, yeah. So, but then the point is, even with the pulmonary pump, the inhaler that we talked about, so that is designed very carefully. So it's more like what should be the particle size, how the flow rate is going to be. So they ask you to inhale very rapidly when you are taking it, so that mostly it will go to the to the other side. So in terms of toxicity, so as I told you, biology is never 0 to 100%, 0 or 100%. So still you can have some remaining in the other side. But then at least when the design is happening, they really try that when you have, so that's why they will give you all these instructions that how strongly you need to inhale. Or the pump design, like when you are pressing it, then what should be the speed of that, the delivery that is happening. So those are all taken into consideration, but that's why these are always developing. These are always in development. That how to make it less toxic and more efficacious. Okay, now about the last question. So try to think about what can be some of the what you would like. So you like in every way we already talked about. You are taking multiple doses of drug and all, but then is something coming to your brain that if this is available, then my life is going to be easier and much more convenient. Hmm? No frequent dosing and hmm? Non-invasive. Yes. Yeah, so mostly yes, patients are going to say this, right? That more patient compliance, and also we are right now because you are younger. I am also relatively young, so that's why we are thinking in terms of when we have fever and something, and then we are taking medicine only when we are sick. But then there are going to be some diseases where these are chronic disease. That means someone needs to take medicines throughout their life, right? So in such cases, for them, the most important thing is how I am limiting that every morning I am not taking a medicine. So for those chronic cases. So that's why mostly how can we design a system which we are going to just put somewhere and then the release is going to happen throughout. So the ideal goal is that whenever there is a disease, you are just putting something, implanting or putting a patch or something. But then whenever there is a disease, then only the release is happening, and then it will stop when there is no disease. So that is kind of the dream. But then at least right now, I mean that is something that also will briefly catch upon on one of the slides. But then right now, at least for chronic diseases and all, that can we at least put something which is control, which is releasing it very slowly. So as we talked about that therapeutic window, so it's going up and down. But then what if we are creating a system that will very slowly increase, but then somehow it will remain in your therapeutic window for a very long time. Okay. So so those are some of the designs that we are going to talk about now. So I know that I have a lot of slides and we don't have as much time, so we'll see how we are going to manage. These are called control release systems or sustained release systems. So some of these, so now we are not talking about your tablet or anything. We are talking about what are all the what are all the invisible material that goes into the design part. Okay. So that means we want that something like there is a predominant rate of release of your drug over an extended period, which is therapeutic, but also it's not toxic. Okay. So that means you have your drug encapsulated in a system, and then it's slowly getting released. So that's why, oh, we have a diagram. So that's why when we are looking at your concentration of the drug in blood versus time, then what is happening is, okay, someone was asking this question. It's called subtherapeutic level, okay, so a toxic level. But then instead of you are having this profile, somehow your concentration is remaining in your therapeutic window for very long time. So that is something which has been tried for a over a few years, and then we are going to talk about some of this that has already happened. So how this process can happen is, maybe I am going to give you some of these bigger terms, but the concept remains very similar, which is you have a kind of a reservoir system. You have all your drug particle. Either your drug particle is diffusing. Now again, coming back to the meaning of diffusion, is just that inside your capsule you have more concentration of your drug. So that's why, and outside you don't have any concentration. So that's why there is a concentration difference and they are coming out. The second one is, whatever reservoir you created, that is getting degraded slowly from the outside. So when that degradation is happening inside your body, your drug is getting released. Okay. Then the third one is swelling. So swelling we already briefly talked about, that you have water and then your material is getting swelled and you have more porous, more pores, and then your drug is coming out. Then the last one is osmosis. So that we will also cover in one of these proper slides. Like you are creating this semi-permeable membranes so that you are attracting the solvent, and then when it's swelling off, then your drug is getting out. So this is just more schematic of the same thing, that either you have a protective barrier through which it needs to either degrade, or through which it needs to your drug molecule need to penetrate out. But I mean, this is the same as the other one. But now, so we'll talk about some of these materials that people already use. So when I told you that you have a reservoir system, so people usually make these polymers or make these other type of materials, and then you now know that you have a chemical compound. You want that chemical compound to sit inside something. So that means you will need either some chemistry so that they are conjugated to your matrix, or you want some physical interaction, either electrostatically, like some covalently, so they are interacting with your reservoir. That's why they are there. But then when you have this external environment changing, then that bond is breaking off, and then your drug is getting released. So so this is something, so which is the hydrogel. So you have polymers, and then these polymers, when they will see water, then they are going to form a gel type of substance. And then when that gel formation is happening, there is going to be some swelling, so that your drug molecule is getting out. Okay. So the osmotic one, so in very simple terms, so you have a semi-permeable membrane on the outside, okay, and you have you have this like your water can go, like when you are in your biological environment, there is going to be surrounding fluid through which your water is going to come in, and then there is this polymeric push. So there is a polymeric compartment which is going to swell once you have water through the osmotic membrane. And then once it's swelling, then you have your drug reservoir on the other side. So that's why when the swelling is happening, and also you have this orifice through which your drug is getting out. Is it very difficult? Should I repeat again? Okay. So now, supremely heavy slide, but then you can design multiple compartments. So based on how many what are all the parameters that you want to include for your drug to be released through that orifice. So you have that smaller orifice in the at the top. So in this one, we just have a single compartment. That means you have a semi-permeable membrane, you have drugs inside, and then you have some of these molecules which are called osmogens, once you will have the water in, then they are going to interact with the water and then they were going to push the drug. So, and then you have this outlet through which your drug is going to get out. Then there is one two compartment system in which you have your semi-permeable membrane, you have a divider. So your osmotic agent is here, but then your drug molecule is on the other side, and then your orifice is there, on the side of the drug. So then what will happen is you are getting water inside, and because of the osmotic pressure, these are going to expand, and then they are going to move this barrier. So because this barrier is moving, then your drug is getting released on the outside. So, and then this is again the multi-compartment system. So you can build that. Why you are designing all of this? Also, it has to do with how expensive something can get. So let's say you want to just use water instead of all of the osmotic reagents and all. But basically, so these are some of these processes through which like scientists have already used this, and this has been implemented for many of these chronic diseases. And as someone told that if you want to have completely non-invasive, so you are just putting a skin patch and that is going to somehow release something very slowly. So even diabetes, some people will say that what if you have a patch, right? And then whenever you have glucose, high glucose level, then your insulin is getting released.
We can also use more than one type of drug. Like if we have to treat two things simultaneously, can we use that?
You can. You can use multiple type of drugs. So I don't have a very good example in my mind, but I know that that has been tried. Why do you have compartments? So okay, so right now what I am showing you is some of the approaches that already exist, right? That how people are designing, that we know that this is one of the criteria. How it is going to affect the drug is in terms of release kinetics. So how much you are loading. First is you have to think about how much you can load. So if you have a one compartment model, then probably you can put a lot of drug throughout that compartment. But when you are changing your compartment, then probably you can stuff less of it. But then here, it's more controlled, right? When you are adding more and more compartments, then you are you are putting some sequential processes in action. So that's why the release is going to be more controlled, more sustained. But in that case, probably not. You may have initially really burst release. That means you are getting a very jump, higher peak.
So in the case when we are using two drugs, how do we determine which one will act first? Is it based on the solubility?
Which one is going to act first?
In case we have two drugs which are used to treat two different problems in the body, so how do we determine which one will act first?
Okay, so it doesn't happen in that way. So most of the cancer drug that we have, so most of the times cancer is treated with dual action. So dual targeting, dual action, that is very common. So when you are taking, so just to address cancer, you can take two drugs, but then their mechanism of action can be different. So it's like eventually they are getting you synergistic action. So it doesn't have to be which one is first, which one is the second, as long as you are getting a better combined effect. Yeah. No, so that's why you are already creating this barrier so that only your drug is releasing from through this orifice.
So how is this better than the single compartment?
It's
In both only one drug is getting released.
no, in this one your osmogen. So here somehow we just have the drug. It shows drug, but you still need to have some osmotic reagent, which is already dispersed. So they can also get released. Yeah. So I think this is the case when you are not just relying on the biological environment. So that's why your semi-permeable membrane is already here. So let's say you are putting it at a location where you don't have the much of the fluid. Okay. Should we move on?
Are the osmotic agents toxic to the body? Is that why they're not being released?
It can be, but also you want that osmotic agent to first of all, yes, it can be. Even if it is not, you still want them so that it's doing its job, which is to swell and then push out the drug, right?
What happens to this osmotic agent after the drug is delivered?
So, I mean, with the entire system, what is going to happen with the entire
After
thing?
the drug delivery is completed.
After the drug delivery is completed, so that's why we want to create this biodegradable material so that it will slowly degrade out. So that is the goal. But why I was showing you this is because you all know what is osmosis, right? And then have you ever thought that in drug delivery people are going to use osmosis? So they are using this polymeric polymers you must have already heard about in many ways. But then have you ever thought that polymers people are like, you know, how that is important for drug delivery? So okay, any other questions?
What diseases are currently being treated
With this, so there are some of these, like, you know, chronic diseases that are being treated. So one of the example is during menopause. So that is one of the best example where they just have a patch and then that is the drug is going to released. So then there is another called multiple sclerosis. So once you have it, then you are going to, like, you know, that is going to continue for longer time. So some of these cases you have this like, you know, you can have these antibodies and all inflammatory agents as the drug molecule which is going to be treated. Okay, so now we talked about how the drug is going to be released, all the engineering principles, but also even before we talked about when it should be released, right? That you don't want it to be released at, like, you know, at all the time. So one is, so these are called, like, only when you have external stimuli that is present, then only you want that release to happen. So in biological environment, one of the external stimuli we already talked about is the pH, but then there can be multiple other type of stimuli. But again, this is what is happening is you have your reservoir and your drug system. Then once it reaches your target site, then there is going to be some stimulus because your disease environment is going to be different. Either temperature is high or you have some type of chemical compounds through which that is going to trigger the release of your drug. And then so and because the degradation happening or swelling happening or some of this physical phenomena is happening, you are eventually going to get some controlled release. So these are some of these probably very small text. These are some of these different stimuli that has people have are taking advantage of that to release drugs. So the first one we talked about pH, temperature. So there are sometimes even with mild change in temperature, can you make a polymer which is very sensitive to temperature, and only when there is rise in temperature, then some of these bonds are going to break inside. And then redox responsive, which is more like there are some of these cysteine double bonds present, and then you are kind of, like, you know, using that environment to break the bonds. Enzyme, so many places has, like, for example, cancer has really different type of enzymatic environment compared to your healthy environment. It has overexpression of a lot of different enzymes. So one of these very cool application I will tell you, which is used for like detection. So what happens is I apologize that I have not introduced you to nanoparticulate, but we'll just talk about it. So basically you have a bigger particle which contains really small tiny particles inside it. But then those are connected using some of these, like, you know, some of these bonds which can be cleaved by very specific enzymes that are present in cancer environment. So first you administer those big particles, then once it will go to cancer environment, these big particles, they can very specifically go to cancer, not to the healthy, because of one of these aspect. In cancer, your blood vessel is forming very quickly. That's why they don't have time to form really sophisticated and proper blood vessel. So there are a lot of gaps inside. So that, and which your healthy environment doesn't have. That's why utilizing those gaps, your larger particles can go accumulate in tumor. Tumor has these specific enzymes which are going to cleave the bricks. Once the bond is cleaved, then those tiny particles are going to be released. Those tiny particles are less than 5 nanometer, and as I told you already, our kidney is going to pass everything that is less than 5 nanometer. Now you will have, so you will just take urine samples and then you will detect that how many particles, smaller particles you are going to get. If you are getting your smaller particles, that means there is cancer. Okay, so that is something that is like that, so not necessarily these are in clinic. Most of the things now I am saying is some of them are just in like in the research stage, but then you can see how much engineering is already involved, engineering plus biology. And then we have this glucose responsive, which which has always been a dream because of the diabetes, that if you have higher glucose, then only will have some release, otherwise there is not going to be release. And then whatever I am saying is the exogenous, the light responsive. So you are exciting light from outside. So similar systems can be ultrasound. So you have a system, but when you are like you somehow put your particles in the area, but then when you are giving some ultrasonic wave, then only your particle is going to release your drug. Another is magnetic field. So can you use somehow magnetic field to take your particles to the location? Very recently, like there are some of these papers where they are taking your cell or taking some of these therapeutic molecules using magnetic field to the location, and then they actuate using the magnetic field so that they are just rupturing all your cancer environment. So and then this is you remember. You remember the like when we talked about the oral absorption and all. So you have your drug, it needs to get absorbed. We talked about through the small intestine. So let's say what we are the design that is happening here, that instead of entirely solubilizing and then making the active ingredients separate from the system, you still have your active ingredient in these polymeric or particle systems. But then what this is going to do is, instead of getting through the small intestine, these big particles are going to attach to the one side of your small intestine. And then what can happen is slowly it's going to, because so this is called mucus adhesion, because your small intestine has these mucus layers, which is like mucus, you all know, which is the viscous jelly like substance. And then once it is adhered, then slowly your drug is getting absorbed to the blood vessel. So basically, even if you have your oral delivery, you are still finding a way to do controlled release. Okay, so then we'll talk about how many of you know what nanoparticle is, and in which context you know it. Oh, someone already talked about this. Okay, so basically, then they have already talked about that you have different type of nanoparticles, and depending on the size and all, you can load. So based on, so I just have this one slide and the next slide, so we won't go deeper then. So basically, you can load many of your like drug, and then depending on how you are using the cell, so it can either target, you can get your targeted delivery. So I actually thought that you are going to talk about nanoparticle in terms of materials, not in terms of biology, and we will come back why I told you that. So, like nanoparticle systems. So there are many types of nanoparticle drug delivery systems nowadays. So and then kind of you have more or less the same like same construct, that you want your drug to be inside, and then you have something that is already covering your drug, but then on the outside it's forming these surface layers, which is going to be easier to get like to penetrate your cell wall or multiple different barrier, like kind of we talked about immune system, like if you can put something on the surface which is going to escape immune system. Are all this covered last lecture? Okay. So so you will see that you have this drug molecule. This is the surface modifier, right? So what happens is, so when I told about this immune system, so they are going to, their job is anything that is coming as a foreign particle, their job is just to eliminate it, right? Take, not eliminate as in they can literally digest it, I mean engulf it. Okay, so and then that interaction also can happen based on the like charge based interaction, because our cell walls are usually negatively charged, and then our like so if they will see a highly cationic surface, they can take it. But more or less what it has been seen is that there can be, like if you have charged particles, then they can get cleared by the immune system very easily. And then there are a lot of, also another thing happens, which is you are putting something in your blood. Blood already has a lot of these proteins that is suspended. That's why it's so viscous. So if you have a charged particle, that means your either cationic or anionic type of proteins that is present in your blood, that is going to cover your entire system, right? Your entire, like your nanoparticle. So eventually they can either get engulfed by the immune system, or they will get into the liver. So one of the way that people figured out that what if we will make our surface as neutrally charged. So for a long time it was found that if you are just putting some polymers which are hydrophilic and neutral, they are not going to be absorbed by a lot of proteins, and also somehow they can also escape the immune system. So, but then now that notion is slowly changing, because very interestingly our body actually learns. So once a body figured out that this is one of the new foreign particle over, like administration are done. Okay, so I just have one more slide, but I was this is very extra things that I was telling you. So once your body will understand that, it can actually educate the immune system that this is something new, now create antibodies against that. So right now what is happening is, whatever that molecule, so this is a PEG, polyethylene glycol, that people started using, but now our body is making antibodies against PEG when you have repeat administration. And then, but more importantly, I think this part someone must have already talked to you about the targeting agents, that you can still have targeting agents which are going to target particular location. Okay, so this is the very last approach that I will say, which is now the futuristic approach. So can you, so that's what I told you, that the ideal goal should be you just put something inside, then that is already a very intelligent self-learning process, which is saying that I will check all this environment, external environment, and only when needed. So it will be able to do some logic processing, that if my A is high, B is low, C is high, then only I am going to release my drug. And then when I will see that the environment is becoming normal, then I will stop releasing it. So this type of logic processing, that I will talk more about it in my next lecture. But then now we'll just summarize, we'll wrap up. So what are all the fields now do you think that are involved in this? Like designing your drug delivery system. We started with chemistry and biology. Now what are all the disciplines you can, can come into your mind? Biotechnology, pharmaceutics, nanotechnology, mathematics, anyone? Or computer sciences? Yeah, computation. Okay. And also, I think you are leaving behind because you are not aware of those. So material science, we talked a lot about different material, which we did not because of the nanoparticle side. Polymers, right? Okay, so all the design principles that I talked about, that is actually coming from these two labs. So this is a faculty at MIT, and the other one is at UT Austin. So both the places that I have been to. So this is from my PhD time and postdoc time. So basically, so all the hydrogel type of system I told you, that is that are modeled by and designed by Dr. Peppers. So he has all these equations, all the pharmaceutical industry, they still use those. So most of the content that I covered, because I took his class, so most of the content that I covered is actually coming from his lectures. Then the other person is Bob Langer. So he is someone that who is the very first chemical engineer. Both of them are chemical engineers, by the way. So they did their PhD together at MIT. So this is the very first chemical engineer who had to fought for to say that we can use nanoparticle in biology. Because until he is a chemical engineer, until that time nanoparticle has been used in materials outside, and then people were very skeptical about how you are putting this particle inside a biological system. So he had to struggle a lot, but right now he has multiple companies around that, Boston area, a lot of startups. He has a huge army of postdocs who are spread all around. I am not his postdoc, so that's why I am not advertising. So don't think that. But did you hear about the Moderna vaccine that came during COVID? So the nanoparticle system that we talked about, or someone talked about last time, so that nanoparticle can carry this mRNA, which was used for COVID, to treat as a COVID vaccine. This is the very first time that got clinically approved. That is coming from his lab only. So now you can see that how all these concepts, also the osmotic, so that very simple, this was one of his first work, and Moderna vaccine is one of the recent works, but then they work on multiple other things. But yes, so basically these are some of these definitions that I thought that I will point out what drug delivery is. So but I mean, we don't have to go through it. So drug delivery, as I told you, that the journey which starts from the injection site to the delivery site, route of administration is through which way you are going to take it so that you are getting very rapid action. Then the third one is pharmacokinetics. That means what your body is going to do to the drug. Okay. Then the last one we talked, the other one we talked about, that is only one of the approach. There are many, many approaches. So the control, like how can we control the rate in which your drug is going to be released into the system? And then for a very long time without inducing any toxicity but having an effect. And then the interdisciplinary nature. So I pointed out chemistry, biology, material science, engineering, pharmaceutics. But I think some of you actually talked about computational work, which I did not put it, but it's highly relevant now with all the lipid nanoparticles that I am telling. So those are completely, people are using high-end AI/ML to make those possible. But yes, so I hope that by now you can answer all these questions again. So can we do that in 2 minutes and then we'll wrap up. So this is the final. So I think the first one we already did, and then the major pharmacokinetics parameter. Can someone? Yeah, perfect. And then explain a few design criteria. So some of these concepts that we talked about for the prolonged, like the control release. Mm-hmm. Yeah. And then the matrix one. Mm-hmm. Hydrogel based systems. Okay. Encapsulation. Nanoparticle systems. Okay, perfect. I think we have covered everything, and then that was the very important learning point, because again I will reiterate that I come from chemical engineering. Now I am in chemical engineering. I don't know what you thought. You didn't know, of course, that I am from chemical engineering background, but I don't know if you thought that I am going to give this lecture about drug delivery, right? But then you will see that the Bob Langer, so he is the one who is the first chemical engineer who started this drug delivery field as a nanoparticle system, and then most of the chemical engineering departments now, they at least have someone or other who is working on drug delivery. Okay, so we'll stop now if you don't have anything else. And then the next lecture, we will talk more about synthetic biology and how to use these logic gate based decisions. Okay, thank you so much.
Such a complex biological topic like drug delivery in our body to something very simple so elegantly. So I would like to thank you for taking your time to come take lectures for us. Thank you so much.